All right. Welcome, everybody, to Standing for Truth. I'm your host, Donny Budinsky, and here at Standing for Truth, we uphold the authority of Scripture from the very first verse while examining the scientific evidence surrounding the origins debate. Today, I'm joined by a very special guest, Dr. J.L. Wile, a nuclear chemist and respected science educator, for an engaging seminar on creation versus evolution. Together, we'll explore key scientific questions about origins, examine common evolutionary claims, and consider evidence that supports the biblical account of creation. This is a thoughtful and informative discussion you won't want to miss. Now, this isn't Dr. J's first time here. You were here a month or two ago, brother, for a program titled Why This Nuclear Chemist Believes in a Young Earth. And I had a lot of fun. That was comprehensive. So I'm glad to see that you had enough fun to be willing to hang out with me again for another couple hours. So Dr. J, how have you been, brother?
Oh, well, I've actually been better. I just got a herniated disc.
Oh,
So I'm having a lot of back pain, but
sorry to hear that.
it's all right. Other than that, I'm doing great though.
Well, I'll be praying for a quick recovery. I know that can be tough in light of what we do with creation apologetics, teaching. I know you do a lot of teaching. So I'll be praying for a quick recovery. And I appreciate you doing this again, Dr. J. So for anybody just joining us in the live chat tonight, creation versus evolution. And we are going to be thorough. I know Dr. J is going to be touching on some of my favorite topics. So stay tuned. And if you're a critic in the chat and you have some objections or challenges, send those in. As you know, we like to be interactive with these programs and engage your questions and comments. So before we hand it to Dr. J for his presentation, I do want to give him a formal introduction for anybody who might be unfamiliar with who he is. Dr. J.L. Wile is a nuclear chemist, educator, and author with a PhD in nuclear chemistry from the University of Rochester. He has conducted research funded by the Department of Energy and the National Science Foundation and has published more than 30 peer-reviewed scientific papers. Dr. Wile is widely known in the homeschool community as the author of the Discovering Design with Science series and the founder of Berean Builders Publishing, where he develops rigorous Christ-centered science curricula used by thousands of families worldwide. That's awesome. Praise the Lord. He is a respected speaker and teacher dedicated to making science accessible while defending a biblical worldview. I may have said this last time. In other words, you're more than qualified to be speaking on these topics, Dr. J. So, okay, I think we're ready for your presentation. Again, one last reminder to the audience, we will be jumping into a Q&A and informal discussion once Dr. J is done with his talk. So please send in your questions and your challenges. Okay, I can get your slides up if you're good to go.
Okay, very good. Yeah. So this is creation versus evolution. And in this kind of subject, you got to concentrate on something. You can't just go all in because there's just too much out there. So in this particular talk, I'm talking about DNA, and that kind of leads to a discussion of vestigial organs, which you'll see how that happens in a minute. So if you don't know, junk DNA has been a fundamental concept in evolution for quite some time. I was told confidently as fact when I was at university that 98% of the human genome is composed of useless junk. And from an evolutionary standpoint, this really makes sense because, first of all, evolution works on a random, it's like a random number generator. The mutations are random. Now, if something good happens, natural selection selects that. So it's guided in some way by natural selection, but the fundamental underlying mechanism is random. Well, when you're randomly trying to solve a problem, you're going to come up with a lot of dead ends. And so it makes sense that the more evolved an organism is, the more evolutionary dead ends there should be it to get to where the organism is now. And those evolutionary dead ends should be at least partially shown in the genome. So it makes a sense that a good fraction of a highly evolved organism would have all junk or mostly junk DNA. And the good thing for evolution is since that junk isn't being used, it can mutate like crazy because it's not being used. So mutations aren't going to affect the survivability of the organism because we're mutating stuff that that isn't used. And so that becomes the source of how mutations will provide new innovative genes and so forth. So junk DNA is a twofold thing. It's a representative of all the evolutionary dead ends, but it also is a source for all the innovative evolutionary steps that are about to come. And so it's, like I said, it's been around since the 70s, really, as a fundamental concept in evolution. Dr. John C. Advise gives us what used to be the scientific consensus on this. So he wrote a book in 2010 entitled Inside the Human Genome, A Case for Non-Intelligent Design. So obviously the book's title tells you what he's trying to say. When you look at the human genome, there's a lot of really bad stuff in there that a designer wouldn't put in. And of course, he highlights junk DNA as the major thing that he's talking about. He says the vast majority of human DNA exists not as functional gene regions of any sort, but instead consists of various classes of repetitive DNA sequences, including the decomposing corpses of deceased structural genes. And I love that phrase, the decomposing corpses of deceased structural genes. But that's exactly what is supposed to have produced this junk DNA. You had a gene that at one time was being used, but then either the organism didn't need it anymore or it mutated to the point where it couldn't be used anymore. So it really is a decomposing corpse of a once useful gene. And like I said, this was the scientific consensus. Even when I was at university, this was exactly what was taught to us. And it was a great evidence for evolution. All right. But of course, now we know that's completely false. We know that the vast majority of the human genome is functional. This started back in 2012 when a very large scientific initiative called ENCODE analyzed 1,640 different data sets generated for 147 different human cell types. Now, you might not realize you've got a lot of different cell types in your body. And in fact, if we start from exactly when fertilization occurred until your death, you have a lot more than 200 different human cell types. And so this study looked at 147 of them. So like skin cells, nerve cells, heart muscle cells, that kind of thing. So they looked at 147 different human cell types, and they wanted to see what parts of the genome those cells used. What parts they actually went into the DNA opened up and the DNA was read by the cell and then brought out of the nucleus. And so if the cell is reading the DNA and bringing that information out of the nucleus, then it must be using that DNA in some way. And of course, they published this and they found, as the journal Nature declared, among the many important results, there is one that stands out above them all. More than 80% of the human genome's components have now been assigned at least one biochemical function. Now, when Nature published this, and there were actually several other journals that published other papers from ENCODE all at roughly the same time, there was a lot of buzz in the evolutionary science world that this was really wrong. That ENCODE had too broadly defined what function is. And so it's not 80, there's no way it's 80%. Their idea of what functionality is wrong. But as time has gone on, because, it's been 13, 14 years, as time has gone on, we found that their definition of function is accurate. Because in the end, the cells are using these portions of the DNA to actually get information. And if they're using that portion of the DNA to get information, then that is obviously a useful part of the DNA. And since 2012, that number has increased significantly. In 2020, 8 more studies were published by the same group, and they found even more functional DNA regions. By 2021, the journal Genome Biology and Evolution, that's the specialty, genome biology and evolution. This journal declared, quote, the days of junk DNA are over. So from the early 70s up until, before, shortly after 2012, but definitely by 2021, junk DNA was considered a scientific fact. And it was not only evidence for evolution, but it was the source for genetic evolutionary innovation. And we now know that's just false. The days of junk DNA are over. And this is important in many ways because, first of all, where's suddenly this thing that's supposed to be great evidence for evolution has disappeared. And you see this throughout the history of evolution. Here's this great piece of evidence for evolution. People talked about it and said, oh, this is great. And then 10, 20, 30 years later, it's completely debunked. And you don't hear about it because they've come up with other things to take its place. And this is one reason you can tell evolutionary theory is not a very good theory. Because in the end, things that are considered evidence for it ends up not being evidence later on. A good theory, the evidence stands throughout the time throughout history. But for evolution, evidence appears and disappears and gets replaced by something else. More importantly, however, it's a recent example of the scientific consensus being dramatically wrong. I mean, not just a little bit wrong, dramatically wrong. And the problem is when most people who aren't very knowledgeable about science or knowledgeable even about the scientific method look at the scientific consensus, they say, well, the scientific consensus is obviously the most reasonable thing to believe because I don't know all of this stuff about science. So how in the world am I going to know what's right or wrong when it comes to evolution, creation, that sort of thing? I'll just look at the scientific consensus because the scientific consensus is reliable, right? Wrong. The scientific consensus is wrong, has been wrong throughout history. And this is the nature of science. This isn't bad. Science is very good as long as we understand that the scientific consensus is always tentative. And if we don't understand that scientific consensus is tentative, if we go with this idea that, well, the majority of scientists can't be wrong on this, even the vast majority of scientists can't be wrong on this, that's not a reasonable scientific statement. And certainly when you look through history, you find it's a statement that's been falsified time and time again. This is just one of the more recent examples. Because remember, it was the scientific consensus since the 70s that 98% of the human genome is now is useless. We now know that's absolutely false. And so whenever we hear people say, well, you're arguing against the vast majority of scientists, how can you possibly be right? The answer is because the vast majority of scientists are often quite wrong. And all you have to do is look through history to see examples of this. This is simply the most recent of those examples. Now, of course, evolutionists were a surprise. I'm not sure I can pronounce this guy's name, so I won't. But this scientist here, this is his picture. There's his name. He was part of the ENCODE team. And when the results were published, he obviously knew the results ahead of time, but he waited till they were published to say anything. He said, I don't think anyone would have anticipated even close to the amount of sequence that the ENCODE uncovered that looks like it has functional importance. He says he doesn't think anyone would have anticipated anything close to that. However, there was a whole group of scientists who did. It was the creationists and the intelligent design advocates. They not only anticipated this result, they predicted it quite a long time ago. So, for example, and this is just one example of many, Dr. David DeWitt made this video almost 10 years before the ENCODE project published its result. If you can't read the title is Junk DNA is Not Junk. And this represented the creationist and intelligent design consensus, because in the end, we see DNA as being a highly designed molecule. And just like anything that's highly designed, a few things could go wrong and it could still work. I mean, think of your car, your car's engine and all the electronics and everything, all very well designed. And, I've got a car with character. There are certain things like sometimes the heat will be on and then it'll just kick off and I have to turn it off and turn it back on again to get it to go. So that's something wrong, but it doesn't really affect me too much. And it certainly doesn't affect how I drive from place to place. So I've got this highly designed thing called this automobile. And there are a few things wrong with it, but I can still get around in it. However, if there were a lot of things wrong with it, the car simply wouldn't work. It wouldn't run. And so when you have something highly designed, it won't work when there's a lot of problems going on. And so we said, look, DNA works great. So the idea of junk DNA doesn't make sense. There can be a little bit of DNA that's been broken, and I think we can find examples of that in the human genome where something that was functional has been broken. The gulo gene, for example, which is part of the vitamin C synthesis. In some mammals, that gene works and they don't have to take in vitamin C from their diet. They make their own vitamin C. We can't do that because one of the genes in that the gulo gene, one of the genes in the process of making vitamin C has been broken. So we can see some examples of genes that have been broken, but that's a tiny fraction of the total. Exactly what you would expect from a design structure. You can have some broken parts, but if you had too many broken parts, like 98% broken, it wouldn't work. So the creationist and the intelligent design community, they're two completely separate communities, but nevertheless, they both agree, look, junk DNA can't be the majority of the human genome. And of course, we now know, in fact, very little is of the human genome is non-functional. And this is actually very bad for evolutionary theory. And there's at least one honest evolutionist who says this. His name is Dr. Dan Grauer. He's a professor of biology at the University of Houston. And he puts it quite succinctly. If ENCODE is right, then evolution is wrong. And remember, ENCODE is a group that said there's more than 80% of the genome that's functional. And he's absolutely right. He really is. This is one case where I really strongly agree with an evolutionist. If ENCODE is right, then evolution is wrong. Why? Think about it. Natural selection needs to be able to weed out all the harmful mutations that occur. If it doesn't weed out those harmful mutations, then they build up over time and something really bad happens to the entire population. So evolution needs disposable organisms in order to weed out those harmful mutations. So all Dan Grauer did in a 2017 scientific paper, he made a quick model of the rate of mutations that we experience in the human genome and how many children every family would have to have in order to produce enough disposable organisms to weed out those harmful mutations. And he said, based on the number of children people around the world have and how the human population's, children's portion grows, the very most of that the human genome can be functional is 15%. And I read his paper, and I'm not an expert in this field, so and parts of his model were a little simplistic, but nevertheless, he's making a really good point. He's saying, look, we've got 3 billion units of information. They're called base pairs. So the human genome has more than 3 billion, it's actually something like 3.3 billion base pairs. Now, if you don't need to know what a base pair is, all you have to know is that's 1 unit of information in the genome. Think about Morse code. Morse code can condense the entire English language into dots and dashes, right? So a dot and a dash. A dot is 1 unit of information. A dash is another. In the genome, a base pair is a unit of information. And so he says, look, we got 3.3 billion base pairs in our genome, and natural selection has to be able to clean up any mutations that occur there. As a result, we have to have a lot of organisms that natural selection can kill to get rid of those mutations. And so based on our mutation rate and based on factors like that, the very most that natural selection can be working on is 15% of the human genome. Well, ENCODE says, no, it's way over 80%. And the Journal of Genome and Biology says, look, the days of junk DNA are over because there's just not much there. So I don't know where Dan Grauer is on this point. Every now and again, I kind of look to see if he's published a new paper. I seriously doubt that the more well accepted the ENCODE results are, the more he will start giving up the idea of evolution. I expect he'll find some
Oh, I think Dr. J may have froze, perhaps.
If the
Dr.
vast
J?
majority of the human genome and the vast majority of most highly, quote unquote, highly evolved animals is actually mostly useful. If that's the case, then there's no way evolutionary theory can survive. Now, we might be able to produce some other theory that can be a materialistic account for origins, but evolutionary theory itself depends on natural selection weeding out harmful mutations. And you can't do that if you've got these huge genomes that are mostly functional. And so he recognizes that, and I applaud him for recognizing that. It'll be interesting to see how far he goes, because in the end, I think he's absolutely right. What ENCODE has done is really destroyed evolutionary theory. Now, most evolutionists aren't going away from it because it's a faith more than it is a scientific theory. But nevertheless, we'll see what happens as time goes on. Now, I want to stick with DNA for a moment because there are a couple other things about DNA that are not only really, really interesting and really, really exciting, but once again, argue against the idea that this is all the result of random processes acted on by natural selection. So I'm sure you've seen numbers like this before if you looked at the creation evolution debate, but DNA has at least 6 million times the storage capacity of our best modern computer systems. Now that's saying something. If I take the best, the 4 gigabyte USB flash drive, DNA is 6 million times better than that when it comes to storage capacity. Now, one of the reasons, this is what most people don't get into, they see numbers like this, but they don't know why. Part of the reason, there's a lot of reasons for this, but part of the reason is that there is this incredible process in molecular genetics called alternative splicing. And this is all, this is amazing. And it's only come to light because we've studied DNA and structure and its use for so long. So this is a little schematic of a stretch of DNA. This stretch of DNA is a gene. Now, a gene is a recipe for making a protein. A protein is a very, very complex molecule. We can't make very big proteins at all in the chemistry lab. If I go in and use our very best equipment and so forth, I can make a protein that's pretty small compared to all the proteins that are out there in organisms, even simple organisms like bacteria. So we can't make it, but the cell can make it because it has this recipe. And this recipe is made up of 2 different kinds of units. There are exons, and those exons are colored in the diagram. And there are introns. The introns are not colored, and they come between exons. So the blue is the first exon in this gene. Then the uncolored space in between is the first intron, and then there's another exon. Now these names actually mean something. The exons are what make it out of the nucleus. Remember, the DNA is in the nucleus. The information in DNA has to get out of the nucleus to a place called the ribosome in order to be used. So we have to have something to carry that message from the nucleus out. Well, the thing that carries that message is RNA. RNA copies the gene, and then it ignores the introns. So it copies the entire gene, but then it says, these introns are just spacers. What I'm going to do is I'm just going to use these exons when I put this together to be taken out of the nucleus. So we're still in the nucleus at this point. RNA has copied DNA, but what it's going to do is it's going to get rid of the introns. That's why they're called introns. They stay in the nucleus. They never leave. The exons are what make it out of the nucleus to be used. Now, of course, because evolutionary theory leads you to all sorts of ridiculous conclusions, Most people who looked at this initially thought those introns were an example of junk DNA. You know, you've got useful stuff, exon 1, and useful stuff, exon 2. And in between, because of all these evolutionary dead ends, we've got junk. And the cell has figured out how to edit out the junk and keep the good stuff. And that was the way introns were first considered, were first thought of. We now know that's utterly false, like most evolutionary ideas. This is just utterly false. What happens is once the RNA looks at these exons and knows what are introns, it says, okay, the cell tells it, okay, here are the proteins I need. And so the RNA splices these exons together in different ways so that different proteins are made. And this is what's called alternative splicing. So the RNA, for example, puts exon 1, exon 2, exon 3, exon 4, exon 5 all together. Remember, it gets rid of the introns, puts them all together. That makes one protein. And we call that in the diagram, that's protein A. However, if the cell needs a different protein, the RNA uses the exact same gene, but only puts exon 1, exon 2, exon 4, and exon 5 together, leaves out exon 3. That produces another completely different, but still a very importantly functional gene. And of course, we can do this again. We can take exon 1, 2, 3, and then skip 4 and put 5 in, and we'll get yet another useful protein. What this shows is that DNA is modular information. The introns tell the cell what the modules are, and the exons are the modules themselves. And as far as we can tell, the introns help the RNA decide where what exons go together and in what order. We don't know that for sure, but that's what it looks like. So anyway, the point is one gene in this picture can make 3 completely different proteins because DNA has been put together in a modular fashion. Now, this is something that took us a long time to figure out because this is a really elegant way to store information. Because think about it, I need a bunch of proteins. How better to store that than to put a whole bunch of proteins in one gene? And that's what we're seeing here. All right. So if you don't get all of that, just understand that one gene, one section of DNA can actually produce a whole bunch of different proteins based on the way the information is put together in these modules. All right. Now, what's incredible about this is that way one gene produces several different proteins. Now, when I was at university, I was taught as scientific fact that one gene made one protein. That was actually considered part of the molecular dogma of genetics at that point. One gene equals one protein. That, of course, isn't true anymore. Now that we understand alternative splicing, we understand one gene, multiple proteins. How many? Well, think about this. The tiger prawn, this simple little shrimp, has one gene that's made up of about 266,000 base pairs. Now, once again, don't worry about what a base pair is. Just think of it as a single unit of information in DNA. So 266,000 single units of information in DNA can be put together in different ways to make up to 21 million different proteins. Think about how elegant that is. I've got 266,000 bits of information. And depending on how the cell uses those bits of information, it can come up with 21 million different functional proteins for the prawn. That's the kind of incredible elegance that we see in genetics. And this kind of stuff is enough to at least convince honest atheists that there's a problem with evolution. One of those honest atheists is Dr. Anthony Flew. Now, I have to say, I was originally an atheist, and when I was a kid, his book, The Presumption of Atheism, was kind of my Bible. Because I thought he was the most brilliant man on the planet, and he made a very strong case for atheism. He was the son of a Methodist minister, but he became an atheist as a teen. He said, quote, "It's just seemed flatly inconsistent to say that the universe was created by an omnipotent and perfectly good being." What he means there is, if God is perfectly good and all powerful, then there shouldn't be any bad things happening. If God is all good and all powerful, why does he let kids die of cancer? Why does he let people who are innocent suffer? Right. And that's a question every Christian has to answer for him or herself. And if you don't know the answer to the question, one of the best books you can read on this is by Alvin Plantinga, and it's called Warranted Christian Belief. And he gives a very good, long-winded, but very good argument for why that's the case. But as a teen, Dr. Flew didn't see that. He couldn't understand it. So he became an atheist. He spent most of the rest of his career arguing for atheism. Like I said, he produced this book, The Presumption of Atheism, in 1976. And the title comes from the fact that he says, quote, "The debate about the existence of God should properly begin from the presumption of atheism. The onus of proof must lie upon the theist." The theist is someone who believes in a God that actually interacts with his creation. And I actually agree with that. And that was one reason I started out as an atheist, because I didn't see evidence for God. That's because I wasn't looking. The more science I learned, the more it became very, very clear that this has all been designed and there must be a God. But I was a fan of Dr. Flew even as a teen. I know I was a nerd. But in the end, I agreed with him that the onus of proof is on the theist. Well, in 2004, he saw the proof, and he wrote this book. All right, so he had spent his career, and he wrote about other things. He was a teacher of philosophy and so forth, professor of philosophy. So he wrote about a lot of things, but everything he wrote was from an atheist perspective, and some of the articles and books he wrote were specifically saying there is no God. So then in 2004, he wrote this book, There Is No God. He crosses out the no and puts A. And the subtitle, How the World's Most Notorious Atheist Changed His Mind. And I think he's right there that he's the most notorious atheist, even though most people haven't heard of him. When you think of atheists, most people think of Richard Dawkins. Compared to someone like Antony Flew or Thomas Nagel, Richard Dawkins is a child. He's popular in the YouTube verse and things like that, but he is not a notorious atheist. He may be a very dislikable man, and he doesn't know science or philosophy very well. But in the end, he's not notorious. Notorious means that he's actually a formidable opponent. And Antony Flew was a formidable opponent to theists until he saw the evidence. What was his evidence? He says this: "What I think the DNA material has done is that it is shown by the almost unbelievable complexity of the arrangements which are needed to produce life, that intelligence must have been involved in getting these extraordinarily diverse elements to work together. It's the enormous complexity of the number of elements and the enormous subtlety with which they work together." And this is really, really, really true. The ways they work together. Sorry. This is really, really true. Like alternative splicing, there are all sorts of subtle things about DNA. Here's another subtle thing that most people don't appreciate. We know that the DNA is a double helix, right? And the reason it has to be a double helix is the nucleotide bases are fairly reactive. That's the reason that the RNA can duplicate them. They're fairly reactive. The problem is, if they were open to the environment, they would eventually react and decay. And so as a result, in order to keep the nucleotide bases more stable, they're in the helix. But they have to be held in the helix with something. They're held together with hydrogen bonds. Hydrogen bonds are fairly weak. They're strong enough to hold the double helix together, but they're fairly weak as bonds. That way, when energy has to be used to open up the DNA to read it, it's not enough energy to destroy all the chemical bonds in the molecule. So the molecule stays together, just the hydrogen bonds, which are the weakest, break. That's another very subtle aspect of DNA. And there's subtlety throughout the genome. And once again, the world's most notorious atheist who believes that the onus of proof has to be on the theist. In the end, he considers that very, very good evidence for the existence of God. And I find it kind of fun and interesting. He was kind of my idol when I was a teen. I started believing in God in my late teens, and ended up being a Christian later on. But in the end, he made the same journey, just in a different way. But once again, it was science who brought him there. Science brought me to a belief in God. Science brought Antony Flew to a belief in God. Now, as far as I know, he never believed in the Christian God, because in the end, he was looking only at science and nothing else. And so he believed in some sort of creator God, but I don't think he had a good concept of what that God is, just a really good designer. But nevertheless, that's what DNA shows you, that there is a God. Another important DNA consideration, and this is something that some people point out in the creation evolution debate, but it isn't pointed out nearly as well as it should be, or nearly as often as it should be. DNA contains the recipes for the cells to make proteins. Proteins are the workhorses of all living things. You have proteins. You have lots and lots of proteins in your body. We don't even know. We can estimate, but we don't even know how many proteins are in the body. But in the end, you've got lots and lots of proteins in your body, and they regulate nearly everything that goes on. So proteins are the workhorse molecules of the body. DNA contains the recipes for the cells to make it. The cell needs to read DNA to get the recipe to make the protein. The way it does that is the recipe is read by RNA, RNA, and then spliced, and then messenger RNA takes it out of the nucleus. Right? It then goes to the ribosome, which is not really an organelle that's in the cell, and that ribosome translates it to a protein. That's the steps. DNA has the information. It's read by RNA, it's spliced, it's sent out of the nucleus, then it goes to the ribosome, it's translated to a protein. But think about it. How is DNA read? And how is it encoded into messenger RNA? Proteins do the work. Because remember, proteins govern almost everything, including reading DNA and encoding it into messenger RNA. That's done with proteins. What's the ribosome made of? The ribosome is made of proteins. But how does the cell get proteins? DNA. So, in order to make proteins, you need DNA. But in order to use DNA to make proteins, you need proteins. What does that sound like? It's the classic chicken and egg problem. Which came first, the chicken or the egg? Which came first, DNA or proteins? Well, the only way we know that cells can make and the only way, even in the lab, we can't make proteins that are very long at all. Nothing close to even the length of the proteins in a lot of the proteins in bacteria. So we don't know how to make them in the lab, right? The only way we know these longer proteins get made is by reading DNA. But the only way we know DNA gets read is through proteins. Now, from an evolutionary standpoint, this is very tough because one of them had to come first in the evolutionary standpoint. From a creationist standpoint, this isn't a problem at all. Of course, this problem goes away if they were both created at the same time for this purpose, right? So in the end, the chicken and egg problem exists for evolutionists. It doesn't exist for creationists at all. And here's how much of a problem this is. Two very well known. Well, the one that's really well known is Jack Szostak. I think Alonzo Ricardo was a grad student who did the actual work. So this is such a problem that original life scientists like Alonzo Ricardo and Jack Szostak says this. Thus, explaining how life began entails a serious paradox. It seems that it takes proteins, as well as the information now stored in DNA, to make proteins. On the other hand, the paradox would disappear if the first organisms did not require proteins at all. So then the rest of the paper is talking about how we might be able to get things that function like proteins, but aren't really proteins. Here's the problem. There's no known organism that does this. Every living organism uses DNA to make proteins and uses proteins to read the DNA. So in the end, what you have to believe in is some unknown organism that used to exist, that left no fossil evidence behind, that didn't use proteins in some unknown process that we haven't figured out. That's where origin of life research is right now. If you don't want to believe in a creator, you have to believe in an unknown process in which an unknown type of organism uses some unknown chemical instead of proteins to get DNA, which can then make proteins. That's a lot of intellectual tumble. What do I want to say? It's a lot of intellectual backflips and handstands to figure out. It makes much more sense to me to get rid of the paradox by simply saying, yeah, they were created at the same time for a purpose. Now, along the lines, this brings us to something else about DNA. So DNA was thought for a long time there was a whole bunch of junk. We now know that there's no such thing or there's very little, if any, junk DNA. Well, along those same lines, we've been told confidently by evolutionists that there are all sorts of junk organs. So one example, for example, is the appendix. So this is a sketch of the human large intestine. At the at the beginning of the large intestine, where it connects with the small intestine, there's this little tube. It's a dead end tube, and it's called the appendix because it looks like something that was added. Right. Now, it looks a little bit like some cecal pouches that exist in other mammals. So when evolutionists looked at this, they said, well, this is obviously some decayed remnant from one of our ancestors' cecal pouches. And this is this is explained in the ironically titled book, The Science of Evolution and the Myth of Creationism, Knowing What's Real and Why It Matters. Here's why it's ironic that she chose that title. Here's what the author says. We have an appendix, a small remnant of prior ancestry species intestinal sac, which not only is of no use to us, but can sometimes kill us when it gets clogged up and infected. What kind of god or other intelligent designer would design organisms with such useless, imperfect, wasteful, and sometimes even harmful physical features? And that was the science. I mean, and already Skybreak is just giving you the scientific consensus. The scientific consensus has been, since the appendix was first analyzed, that it must be just some evolutionary leftover remnant that does nothing, just like all the junk DNA. But of course, we now know the appendix is an incredibly vital organ. And once again, creationists predicted this. In 1988, here's what J. Warwick Glover, a creationist, said. The appendix would be involved in the control of which essential bacteria come to reside in the cecum and colon in neonatal life. As well, it would be involved in the development of systemic tolerance to certain anergetic agents within the alimentary tract, whether they are derived from bacteria, foodstuffs, or even the body's own proteolytic enzymes. So what he's saying is, no, the appendix is really important. And here's the first part. In the control of which essential bacteria come to reside in the cecum and colon in neonatal life. All right. So he said, look, this is clearly a functional organ. He thinks it's mostly focused on neonatal life. That's where he's wrong. It's a functional organ that is needed throughout your whole life. Now, I know what you may be saying. Wait a minute. People can have appendectomies here in the U.S. and no problem. They can live no problem. Well, that's actually not true. They can live no problem as long as they don't get a intestinal infection like Clostridium difficile. And since these intestinal infections are rare, we don't notice them in the population very much. But nevertheless, we now know that J. Warwick Glover's prediction was broadly confirmed. The appendix is an incredibly important organ because it's a hideout for the intestinal bacteria. You've got all sorts of different species, more than 1,000 species of bacteria that live in your intestines and other parts of your body. Not all 1,000 are in the intestine, but you got a lot of them in the intestine. And they do things for you that is absolutely necessary. They digest certain chemicals that you can't digest and would be a problem if they built up in your intestine. They make certain chemicals that you can't make. In exchange, you provide them with food and housing. And so intestinal bacteria are really, really important. Well, when you have an intestinal infection, those intestinal bacteria get wiped out and you have to get them back. Now, if you don't have an appendix, you have to get them back over time by eating lots of foods, lots of different foods, and sampling all the bacteria in your environment. However, if you have an appendix, those bacteria automatically repopulate because the appendix is a place that they can survive during the intestinal infection. The appendix doesn't have the same tissue as the large intestine because its job isn't to absorb stuff. Its job is to have a safe place for these intestinal bacteria to live. So it is like a hideout. And in the end, when your immune system finally clears out the intestinal infection, then these bacteria can come out of the appendix. All right. Now, this is really sad, but because of evolutionary theory, it became standard practice to remove the appendix. So if someone was having any kind of abdominal surgery, it was standard practice to remove the appendix, even if it was healthy, as long as it was convenient to the surgeon. So he or she wouldn't go rooting around to try to find the appendix. But if during the abdominal surgery, the appendix presented itself, the surgeon would just snip it off because evolutionary theory told them it's useless. So why not get rid of it? Because sometimes bad things happen to the appendix. And so might as well just get rid of it. The problem is those people do not recover from intestinal infections nearly as well. Here in the US, the main thing is intestinal infections are just reoccurring, but we've got antibiotics we can use to get rid of the problem so the intestinal infections never become fatal in the US. In the third world, they're fatal. And unfortunately, there were these kind surgeons who would go over to the third world to do surgeries that were necessary. And they were following standard practice, which was inspired by evolutionary theory. They were snipping off appendices there too. The problem is that was a deadly choice for some of their patients because some of their patients then developed an intestinal infection, didn't have access to the antibiotics we have, and died as a result of not being able to have that and those intestinal bacteria repopulate soon enough. So in the end, this is a case where evolutionary theory actually caused death and damage and other things. And this isn't an isolated incident. Evolutionary theory has inspired all sorts of pain and suffering. Here's another example. Tonsils. We were told, evolutionary theory told us that tonsils were evolutionary vestiges. They looked like something from our supposed ancestors. They didn't seem to have a function in humans. And so when they were inflamed, rather than simply giving antibiotics and letting them clear up the infection, the standard practice was to remove them, surgically remove them. After all, they were useless. Why in the world keep something that can get so easily inflamed, that is inflamed now? Why not just cut it out so you don't have to keep taking antibiotics every time the inflammation occurs? All right. That was standard procedure. And so it became standard procedure. I had my tonsils removed. Now, as a kid, I loved it, because I'd never been under anesthesia before. That was kind of fun to be counting and then suddenly fall asleep. But then I got out of school for like a week and I got to eat as much ice cream as I wanted. So I loved having my tonsils removed. The problem is this has led to pain and suffering because we now know that tonsils are infection-fighting organisms. And a study of 1.2 million children comparing those who had their tonsils removed with those who didn't makes this conclusion. Removal of tonsils in childhood was associated with a significantly increased relative risk of later respiratory, allergic, and infectious diseases. Increases in long-term absolute disease risk were considerably larger than changes in risk for the disorders these surgeries aim to treat. So in other words, the long-term risks were significantly higher than the risks that the surgeries were trying to get rid of at the time. And once again, this was inspired by evolutionary theory. Evolutionary theory ended up causing harm because it refused to believe there's a purpose in nature. Someone who believes there's a purpose in nature sees DNA or sees organs and says, look, they're there for a reason. Don't take them out. But in the end, someone who doesn't believe in a purpose in nature is going to say, it's fine, take it out, not doing anything. It's fine for an evolutionary theorist to say, okay, this is a vestigial organism. What's bad is to be so confident in that you end up hurting people. But evolutionary theory has done this time and time again. All right. Now, there's signs that this is changing. I could not believe this when I read it in the journal Biological Theory. This is open access journal. Everybody can go see it. It's called Mind Everywhere, a framework for conceptualizing goal-directedness in biology and other domains. This is actually a 2-part series. The first part is titled something different. And I can get that title if anybody wants it, but you can find it pretty easy because it's published in the same journal, Biological Theory, and it was published before this one. But here is what this article says. What is needed, therefore, is an approach to teleology in biology. If you don't recognize that word, teleology is the study of purpose. Teleology in biology means purpose in biology. It goes on, informed by mentalistic concepts such as intelligence, cognition, and intentionality. Now, they don't say God is it. All they're saying is, look, when we look at biology, there's all sorts of purpose here. And to ignore the purpose is to ignore the science. So what we have to do is figure out a scientific way to look at biology in a purpose-driven theory rather than a random theory, a theory that's based on randomness. And 10 years ago, I'm sure whatever editor of this journal would have been fired for allowing those two articles to be published. But it was published, and actually it's been received fairly well. And I personally think this is actually an answer to a proposal Dr. Thomas Nagel called for in his 2012 book. Dr. Thomas Nagel is one of those notorious atheists who deserves the title. He's an intellectual giant. I disagree with him, but he knows what he's talking about. In 2012, he wrote a book called Mind and Cosmos: Why the Materialist Neo-Darwinian Conception of Nature is Almost Certainly False. And what he says is evolutionary theory cannot explain consciousness, cannot explain the fact that we can know ourselves and know that there's something else out there and all of that. So in the end, we can't explain the evolutionary theory can't explain the development of consciousness. Therefore, it's useless, because consciousness is the main feature of human beings. And if we have some theory of biology that can't explain the origin of human beings, then that theory is worthless. And what he specifically said was, I'm not talking God here, but what I'm saying is we have to have a teleology in biology. And that's what this article is saying. I'm hoping this article starts something revolutionary in biology, because biology needs to get out of this randomness stuff. All of biology does not point to randomness. It points to purpose. And so we have to accept that. And we're going to always be limited in how much biology we can understand if we don't accept that. Okay, that's all I've got. I'm looking forward to questions from the audience. So if Danny can come back and give me some questions, that'd be great.
Dr. J, I've got lots of questions for you, just like part one. And you're so good at answering the questions. You're always informative and up to date, just like your talk. I really enjoyed the flow of that. Junk DNA dismantled, junk DNA is junk science. You discussed other topics related to the junk DNA argument, Dr. J, that a lot of more informed evolutionists will bring up, like the gulo pseudogene. And so perhaps I'll just start with that. Okay, so to clarify, you're saying that as expected or predicted from the Creationist starting point, the vast majority of the DNA appears to be active and therefore functional. But that's not to say there aren't real broken genes due to deleterious mutation accumulation over time. And so are you arguing that the gulo pseudogene, for context for people, and correct me if I'm wrong, basically we have a gene for vitamin C synthesis, but it's broken, so we can't produce our own vitamin C, just like your chimpanzee. So evolutionists will say, oh, we've got those shared genetic mistakes broken in the same way in the same spot.
Yeah.
Therefore, we must have inherited that genetic mistake from a past common ancestor. So the best way to answer that, in your opinion, is that is a true broken gene, but that occurred independently through something like a mutational hotspot, Dr. J?
Yeah. So, and once again, the gulo, it's a pseudogene in our, because it genome, it doesn't work. It works in other ones. So I tend to call it a gene because it's functional in some other ones.
Good point.
But it's one of several. But in the end, if you don't have all of them, then you can't make vitamin C. So the gulo gene is just one of several needed. But nevertheless, we don't have it, so we can't make our own vitamin C. But yeah, in the end, the argument that well, you know, the breaking, the place where the gene is broken is the same in humans and chimpanzees. Therefore, humans must have inherited it. Humans and chimpanzees both must have inherited it from the common ancestor. That's really wrong, because in the end, most genes have mutational hotspots, and most mutations occur the same way. So it's not all that surprising that if our gulo gene got broken, that a lot of organisms that have similar biochemistry are going to have the same breakages as well. It's the way mutations work. So yeah, we accumulated those independently. And please understand that if you look at other supposed pseudogenes that have breakages, we don't have the same ones. So it only works for the gulo gene. And here's what's amazing to me. Let's assume there are no mutational hotspots. So let's assume that it truly is completely random. And so, you know, we accumulated these random mutations. And then according to creationists, chimpanzees accumulated the same random mutations. The evolutionists say the chances of that happening are so mind-bogglingly low, there's no way you can believe that. And even before I knew a lot about the gulo pseudogene, I said, but wait a minute. Evolutionists believe in mind-bogglingly low probability events happening with regularity. In the end, nothing in evolution happens unless you have these mind-bogglingly, ridiculously low probability events happening. The chance of us accumulating the exact same mutations randomly, us and chimpanzees, is significantly higher than the chance of the first protein occurring. So in the end, evolutionists are so used to accepting low probability events. This is an actual high probability event compared to most evolutionary steps.
Well, that's a good argument, because typically when we'll answer the gulo gene argument with mutational hotspots, independent, deleterious mutation accumulation. Well, and wouldn't there be relaxed selection, if that's the right term, anyways? Because as humans, we get vitamin C from our diet. We take vitamin C supplementation. And so wouldn't that leave that gene more prone? And I know taking supplements, that's more so relatively recently, but we still get it in our diet, plants, fruits, so on and so forth. So would that leave that gene more susceptible to breaking down as compared to other genes, Dr. J?
I'm not sure that's the case, because, and I understand where you're coming from. We don't have a lot of negative effects because of this. Only like it only showed up in sailors. And because just the normal human diet has a lot of vitamin C. So I see what you're saying, but I honestly think this is one point where, like Dr. Grauer says, natural selection cannot keep policing all these genes. Now, of course, an evolutionist has to believe all these genes have been around for millions of years. A creationist doesn't. So if we lived for 1 million years, I would agree with Dr. Grauer. Our genome should be decimated. Should not be 98%. But so I think this might be one of those cases where natural selection just couldn't police all the genes. And we've seen this as time goes on, we've had more deleterious mutations. There's one article in the journal Gene, or in the journal, yeah, it is called Gene, that actually shows 2,000 neurological mutations that have occurred in our neurological genes that have reduced the performance of our neurological functions. And his point was, we're probably dumber now than he actually says in the article that if I could bring someone from ancient Athens here, he would be smarter and more emotionally well-adjusted than any of us.
Wow.
Because he doesn't have it. Yeah. So, but anyway, the point is, I think what Dr. Grauer says is true. Natural selection can't possibly police all of these functional genes. So we're seeing all sorts of breakages, but we just haven't been around long enough for the breakages to accumulate to the point where they become catastrophic.
Yeah, that's a really good point. And so, as Dr. Grauer and I've seen others point out, because we've done debates and we've had evolutionary biologists here who will say, okay, at best, creationists, if we consider all, and I think they're wrong on this, but this is their opinion, if we consider all of the functionality in the human genome, including pseudogenes, endogenous retroviruses, ALU sequences, so on and so forth, we'll give you 10, maybe 12%, which I think we understand it's a lot higher than that, because they know really any more than that, especially 20%, 30%, 80%, which is probably more so around the correct number, then would that mean these deleterious mutations that are accumulating every generation, 100 roughly per person per generation, if the majority of the genome is not just neutral junk, well, then those are going to be more deleterious, nearly neutral, slightly deleterious, which means genomes would degenerate faster than if the majority of the genome were junk, Dr. J.
Yeah, and that's Dr. John Sanford's point in his book, Genetic Entropy. Right. He actually quotes a scientific paper. The title is "Why Aren't We All Dead 100 Times Over?" Yeah, because it's cataloging mutation rates and the deleterious mutations that have actually survived natural selection. And he says, if we've been around for millions of years, this should be total decimation by now. So yeah, that's genetic entropy, according to Dr. Sanford.
Okay, and you debunked this next point in your presentation, and we also addressed it in our previous talk program, and I put a reel out because it's so common, but let's leave no stone unturned and do it again, perhaps reiterate, because this is what we constantly see. I had a junk DNA expert, I can't remember his name, but he saw a couple of my programs on junk DNA, and so he emailed me. This gentleman, he's a PhD scientist. I'll look him up after. He just came to mind. He wrote a book on junk DNA defending it. And so even he said that, well, just because ENCODE said that 80% is biochemically active, biochemical activity does not equate function, and most likely due to the low level of transcription that we see, that activity is noise or spurious. So it's not actual, we should not be interpreting that as actual function.
Yeah, so this is the double talk that evolutionists love to give. So ask that same guy why humans have better eyesight than gorillas, but gorillas have better smell than humans. And the answer is gorillas lived in the jungle. The eyesight didn't go too far, so they needed to develop a good sense of smell. And because evolution can't possibly waste energy, it actually allowed the eyes to deteriorate and the sense of smell to increase. Humans went to the plains. Because evolution has to preserve energy, they lost their sense of smell. They lost the acuity and sense of smell, so they could build acuity and eyesight. Ask the evolutionists why a dandelion produces nectar even though it only reproduces asexually. Well, at one time it produced sexually, it lost that, and because evolution can't waste. So, sorry, I'm back up. So we got the ape thing. Evolution can't waste energy. That's why eyesight is better in humans. Sense of smell is better in apes. However, now to explain DNA, evolution is allowed to waste as much energy as it can because it's doing, and it takes a lot of energy to break those hydrogen bonds, read the DNA, put the RNA together, do the splicing, and get it out. That takes a lot of energy. That's probably 30% of all the cell's energy is that, right? Now, it's different if it's a muscle cell uses it, but for the average cell, that's about 30% of its energy. And so it can just waste that, no problem. and there's I think I mentioned him last time, there was an old book by Walter Ramin called The Biotic Message. And what he said was evolution is just this smorgasbord of different possible explanations. And whenever you come up with some observation, you choose that one and that one. They fit together to make this observation. Another observation, we choose that one and that one. We fit them together. In the end, we always can explain away anything we want because we keep making these stories that often contradict each other. Look, does evolution save energy or does it waste energy? If it saves energy, then the ape and human thing makes sense. But the junk DNA thing doesn't. Right. So so in the end, that's my problem. And I have my brother-in-law is one of the most well-known molecular biologists in the world. Can't mention his name because he's not a creationist. Don't want to get him in trouble.
I was just going to ask you what's his name.
His wife is a molecular microscopic what do you call that? Cell biologist, right? So both of them are in on this. And when I ask them about it, they're both evolutionists. And they say, yeah, if the cell's translating it and bringing it out of the nucleus, it's being used. It has to be.
Interesting.
Because
and that's from somebody who holds to the evolutionary paradigm.
Yeah. And his answer is, I don't know. I don't know how this works, but I can tell you if it's being transcribed and brought out of the cell, which is what ENCODE shows, that it's being used. And that's just and I think anybody who's doesn't have a dog in the fight is going to have to, who knows his or her stuff, is going to have to admit that there's just too much energy involved.
Yeah, that's a great response.
And I agree. Some of these I'm sorry to interrupt you, but I agree that some of these transcriptions are very low. They don't happen very often. But remember, it only takes a little bit of some chemicals to make all of the difference in the human body. That's the whole thing about vitamins. Vitamins exist in our body in tiny concentrations because that's what we need, and too much would be bad. So all the low level of transcription tells us is these are molecules that only are needed in small concentrations.
Yeah, that's a really valuable point right there at the end you made. Just because in some instances we have low level transcription, that does not equate, then you're saying, to non-function. It's just in those certain functions, that's all you need is a little bit, versus other functions that are more detrimental, I would imagine, to our health and survival. Well, then you'd have higher levels of transcription. I guess kind of like a car, you'd have an engine, which is used a lot in your car. And then maybe you got your rear windshield wipers, which I rarely use. Doesn't mean it's non-functional. I do use it once in a while.
Yeah, use it when you have to.
It's active in low levels, we could say. Okay, well, I think that's a great response. I did look up the gentleman because I wanted to make sure I was accurately representing his and others' arguments, which you just addressed. His book is titled "What's in Your Genome? 90% of Your Genome is Junk." Dr. Lawrence Moran. So
yeah, Larry Moran. He
okay,
his friends call him Larry. Yeah, I'm not a friend, but his friends call. Well, actually, even his colleagues call him Larry. So, and he has his website is like Sandwalk or something titled like that.
It is, yeah, because he wrote a blog.
Yeah. I think, and once again, the title's wrong. We know that. We know it's not 90% non-functional. But nevertheless, he's going to hold it because he's a pretty ardent evolutionary defender. So he's going to have to come up with these just so stories to explain around. And that's the crazy thing. Think about how quickly they all had to turn, and Moran's one of them. I'm not sure he's completely turned yet. So Moran might not be a good choice, but there are several biologists who staunchly use DNA as strong junk DNA as strong evidence for evolution.
Richard Dawkins, one of them.
Yeah. Now it's not there. So they turn on a dime and say, well, yeah, it's not really that important to evolution after all.
Well, it's funny because when you asked those two questions earlier that were contradictory, I guess the evolutionary answer would just be yes.
Yes.
Yes to both.
Yes to both.
Energetically costly, and we don't need it.
Yeah, and this is why I can't remember the exact book title now, but two guys wrote a book that caused a huge stink in evolutionary circles. They're both evolutionists, and they said this whole evolution thing needs a complete rethink because it's not working. And they had to put in their introduction, look, we're card-carrying atheists. We're not trying to support intelligent design or creations. We're just saying this isn't working. And I can't remember their name. One of them is really weird, like Massimo Pugliani or something. It's a name I can't think of off the top of my head. And they got all sorts of grief. But that's one of the things they said. A theory has to be coherent throughout. I can't choose contradictory explanations for different phenomena, but that's what evolution does. And they admitted that.
Well, I've seen many evolutionary scientists. They'll say, well, you creationists are misrepresenting us. I've had PhD biologists email me with this. We never said all DNA was junk. We never said all non-coding DNA was junk. But have we ever said that they said at all? I mean, sure, maybe one or two, a few instances, but not genome-wide functionality. Dr. J. I'm curious, how would you engage that with an evolutionist who argues?
Yeah, once again, this reminds me so much when Galileo probably didn't do this, but somebody who supported Galileo did. When they dropped a small a light ball and a heavy ball from the top of the tower, and the heavy ball hit just a minute second, a fraction of a second before the light ball. All the people who supported Aristotle said, see, Aristotle was right. Heavy things fall faster than light things. When Aristotle said a heavy thing should fall a lot faster than a light thing. It's the same thing here. They were, we weren't saying all the genome was misfunctional, but they were saying a huge fraction of it, 98% of it was. None of these evolutionists ever said, well, it may not be 98%. Maybe it's only 50%. No. That was an accepted figure. So yeah, they weren't saying all the genome was unfunctional. Only 98% of it wasn't functional. And so to argue that somehow they weren't making this sweeping argument, 98% is pretty much everything.
Right.
And nobody was contradicting that number.
Or even Richard Dawkins. I can't remember the exact quote. I've got the quote somewhere, but he said something that basically sounded like, I mean, most of your non-coding DNA might as well not even be there. Right. It's not even. So then, okay, maybe they said not all of it, but the majority of it would be like me saying, I'm just trying to come up with an illustration, my sock drawer. I mean, it's my sock drawer, but maybe I do have a couple pairs of underwear, t-shirts in there. I mean, it's still my sock. It's still my sock. I didn't say it was all socks, even though it's still my sock drawer. And so they'll follow up and say this. I've seen evolutionary biologists say this. Well, the majority of the genome is junk because of conserved versus non-conserved DNA. A lot of the DNA is not conserved, which they interpret to mean so it can't be important because selection's not preserving it. It's not favoring it. You can't be doing anything too good, which is why it's so prone to mutation accumulation, right? And apparently there's a lot of that kind of DNA between humans and other creatures. Must be non-functional, Dr. J.
Yeah, and see, once again, this is a very static view of the genome. For a science that says the genome can change a lot, because eventually something that gives rise to both apes and humans. So the genome can change a lot. Suddenly they're saying the genome can't change at all. Because in the end, anything that's changing must be unimportant. I think these things that are changing for a reason. And once again, this goes back to purpose in biology. But in the end, everybody's biochemistry is a little different. And your child's biochemistry is a little different from yours. So when parts of the DNA start changing, it could be because it's absolutely necessary. And we know this happens to at least some extent. We haven't done this in human beings, but we've done it in bacteria. Richard Lenski's long-term evolution experiment. They showed how bacteria that couldn't digest citrate under oxygenated conditions, they could do it under unoxygenated conditions, but with the presence of oxygen, they couldn't do it. Actually developed a way to digest citrate with oxygen. And Lenski said, hey, this is a great evolutionary event because we've got this big change occurring. And I agree, it's a big change. It's not as big as he wanted to say because they could do it under one set of circumstances. They just couldn't do it under the circumstances of the experiment. But he said, look, this is a major evolutionary change, and it's clearly following evolutionary rules because it only happened once, and it took like 35,000 generations for it to happen. So clearly, that's an evolutionary event. David Minich comes along and reproduces the experiment and shows that he can make this happen in as little as 12 generations over and over and over again in multiple experiments. And he said, look, this clearly isn't a random event. If it were a random mutation, then in the end, it could only happen every 30,000 generations or so. This is happening in as 12 generations. I can make that happen in just a handful of generations over and over and over again. So clearly, this is a designed mutation. The genome is designed to change for adaptive purposes. So it's not random mutation, it's purposeful mutation. And we know that's the case now. We know there are some mutations that occur, at least in bacteria, that are reproducible and not random, which means in some way the genome is ready to do this. And so the intelligent design people and creationists call those designed mutations. I think evolutionists call them reproducible mutations or something like that. And so all of this changing genome that they see that say it can't possibly be useful, that could all be design mutation, because we know it happens in bacteria. It probably happens in humans too, and every other living organism.
Well, that's a very interesting point and hypothesis, and I think it works. So as a creationist, would we say that we're looking at two things? We're looking at design diversity, that God would have created the original created kinds. Let's take humans with a lot of internal DNA diversity to be called upon for change and adaptation. And so a lot of what evolutionists say are mutations could be designed alleles. But then we also have to think about design mutations, as in a lot of these spots in the genome you're saying are designed through forward thinking, one could say, to mutate in changing environments for adaptive purposes.
Yes. Yeah. And I misnamed him. His name's Scott Minich. Okay. And so if you look him up in the scientific literature, he's a published, but he published a paper that says, and his overall conclusion is, because I could do this, so often and in very few generations, it was a design mutation. And he specifically says that means no new information was added to the genome.
Interesting. Okay.
So we've got this genome that's got all this information, but like the subtlety Dr. Flew talked about, it's hidden and will only appear when called upon by these design mutations. And speaking of names I didn't get right, the book I was talking about, two evolutionists, two card-carrying atheists who said there's a problem here. The title was What Darwin Got Wrong.
What Darwin got wrong.
Jerry Fodor is one of them, and this Massimo guy, who I'm not even going to try and pronounce his last name, is the other one, because I don't want to offend him. What Darwin got wrong. And basically they're saying, look, evolutionary theory can't predict things well, because we don't know which just so story to use until after we've seen things. And there's no coherent method for which just so story to use.
Well, that's really good. I'm looking up the book right now, and I can see there's some Reddit discussions on it. So they're probably a little triggered because you're saying those are proponents of their own side, but they're criticizing aspects of evolutionary, not denying it, but criticizing. Okay, interesting. I like that. I like when we can poke holes in evolutionary theory from their own, and which we can do with their technical literature, because they're the ones that are finding all of these functional roles in endogenous retroviruses, a lot of these pseudogenes, just junk DNA in general. And then obviously they throw in their evolutionary philosophy in the discussion and say, well, maybe these were co-opted or, with vestigial organs you talked about, they'll try and say, well, these were repurposed.
Yeah, so, after first saying they were useless.
Right, yeah. And now they're just
it's repurposed.
But then they'll assert that we never said they were completely useless. We just said that the function would have been different in the common ancestor.
Even though we can quote at least proponents of the theory saying completely useless. I quoted one of them.
Is that historical revisionism at its best, or what? I mean,
yeah,
what people are
to
saying.
me it's compartmentalization. I'm willing to throw that now in a new compartment and kind of forget what I said.
Yeah. And hope everyone else forgets.
Yeah. And I'm not even sure they're lying about it. I mean, you really have to do some intellectual gymnastics to be an evolutionist. And so I think you have to compartmentalize your thinking. And so it's not even clear they're lying about it. I think they put it in the compartment and actually forgotten what they said.
It takes a lot of work to be an evolutionist for sure, to believe you're related to grass and ladybugs. And so there was something I wanted to say based on what you said there. Okay. The design mutations, informed evolutionists, I've seen them in debates, for example, they'll point to this argument. It's called private alleles. And they'll say there's just too many private alleles, which they interpret as mutations in these different lineages. So they'll look at different dogs, so within the canid family or the felids, and they'll say you can tell that these are mutations because they're not shared with everybody, so they must have accumulated since the flood. So they're basically assuming if creation's true and we go back 4,500 years ago, let's look at all these private alleles, which they say are due to mutations. They'll say, and they'll correlate them with some kind of adaptation with the creature, Dr. J. Mm-hmm. And so they'll say these must be mutations that are beneficial in some way, but you can't explain in 4,500 years how all of those could accumulate. But if you're saying, and feel free to expand on this, but if what you're saying is true about the designed allele argument, that a lot of these spots are designed to mutate for adaptive purposes, could that be part of the answer to explaining these alleles that came about?
Oh yeah, and I absolutely agree with every evolutionist that says you can't get the diversity we see after with just the time after the flood if these mutations are random. I absolutely agree with that. There's no question about that. But I do think that we've uncovered a bit of gold here with Scott Minich, and I could do another talk where I talk more about him and his research, but I think he's discovered a bit of gold there that a lot of people are now going to start looking at, and I think that's going to be a big part of the young earth creationist mechanism. But that is one of the things that as a young earth creationist, I would say I can point to examples of evolution that happened really quickly. Like there was this group of wallabies that escaped a Hawaiian zoo. They couldn't find them because Hawaii's big and not easy to navigate. And they found them. I can't remember how many years later, but it was only 40 or 50 years later. They found them in a part of Hawaii that was pretty remote, and they were living off a plant that's poisonous to wallabies, but they were eating it fine. Right. And so in just 40 or 50 years, they evolved enough to start eating this plant that would have killed them, had they eaten it in the zoo. And that's because, in my opinion, the environment put a pressure on them, and some mutation mechanism was activated to make them adapt to being able to eat that plant. And so, but what I'm saying is I can point to certain examples of fast mutation and fast evolution, but I can't explain how we get to a pair of cats that walks off the ark, and now we have everything from Siamese cats to lions, right? I can't explain that with the tools we have now, especially assuming the mutations are random. Yeah. But I can't explain it if the mutations aren't random, which I've always thought. It's just I didn't know if there would be evidence for that, but we have evidence for it now. Only in bacteria. Let's make that clear. We've only been able to uncover this in bacteria because it's really hard to study long-lived creatures. Right. It's hard to study mutation accumulation in long-lived creatures.
Absolutely. Well, and with like mice, for example, you could do genetic knockout experiments. Yeah. Not very ethical in humans, of course. That's difficult to do something like that. Although if evolutionists believe the majority of their genome is junk, go knock some of that out. Let's prove how confident you are in that. And of course they'll say no to that. Okay, that's a good answer. You mentioned bacteria then. I hear put forth frequently these bacteria that apparently evolve the ability, nylon digesting bacteria. They evolve the ability to digest nylon. They wouldn't have that capability, of course, man-made nylon, but could that pre-existing, because I'm thinking about that example you just did with the poison. That's interesting. So could that ability have already been there? Maybe some of the genes present to essentially be turned on?
Yeah, we know some of that. The ability was already there. This is not new. Nylon digesting bacteria are definitely not a modern strain. This is one of these evolutionary stories that gets a lot of press, gets put in books and everything else.
It's a popular one.
It takes a long time to show that was wrong. So there was a study by Sal Cordova and John Sanford published in the peer-reviewed secular scientific literature. Called Nylonase Genes and Proteins, Distribution, Conservation, and Possible Origins. They show that these not specific nylon digesting bacteria, if you look at the ones who don't use this process, the same species, different strain, you see genes that can digest nylon, just not very efficiently, and certainly not efficiently enough for the bacteria to live exclusively on it. But they're already there. All evolution has done is tinkered with those genes to make them more efficient so that these bacteria can exist mostly on nylon. So you might wonder why in the world were nylon genes that could digest nylon already there when there was no nylon? And the answer is nylon has a lot of very similar patterns to actual long polymer molecules that do exist in nature. And so these genes were digesting those polymers. And so it didn't take much tinkering then to be able to adjust the nylon, the actual nylon. And they do a really good literature study that show that over 1,800 organisms have these starter nylon digesting genes that can digest nylon just very inefficiently. 1,800 different organisms. So they didn't just appear in this place where the nylon waste was coming off. They've been around, and all these guys did was their environment either directed mutations or just randomly their environment made these mutations beneficial. But it's not new. And this is really well known. So like I said, I would encourage someone who believes that nonsense to read that paper. And it's not like, once again, it's a peer-reviewed paper. Nylonase genes and proteins, distribution, conservation, and possible origins. S.T. Cordova and J.C. Sanford.
Yes. Yeah, I got it here. I'll put it in the live chat. Yep. That's incredible. So in light of what you said there, I see in the conventional literature, those that have actually attempted to engage or refute the genetic entropy hypothesis, Dr. J, that the most mutations are nearly neutral, they accumulate subject to genetic drift because selection can't see most of them, so they drift and spread. What I see in the conventional literature is a lot of appeals to beneficial mutations and super beneficial mutations. They'll say, yes, they're rare, but when they do occur, and they occur in enough frequency to counterbalance the damage. So they'll admit, okay, yeah, we got a lot of neutral mutations accumulating, but those beneficial mutations will counterbalance the damage. But are they assuming that those are truly non-reductive beneficial mutations, or beneficial mutations that are not just what you're explaining, occurring due to the pre-existing ability to adapt in the first place?
Well, yeah, if there's a pre-existing ability to adapt, like creationists think, then in the end that explains a lot of this stuff. But honestly, I think the super beneficial mutation idea, it doesn't work because we actually see these mutations accumulating in people and in other organisms, and we don't see something counteracting it. So we can show. I mean, I know Sanford did this with a virus, not people, but he had samples of one of the swine flu viruses. H1N1. He had for over 100 years, because there were some explorers frozen in ice who had that virus, and so you can look over time and see how those mutations develop, and they continue to be deleterious, and there's no big beneficial mutation popping up. And you see this like that article I talked about in the journal Gene, 2,000 deleterious mutations in the neurological genes, and we don't see anything big popping up, right? So you can kind of believe that might happen at some point, but I don't have any evidence that it happens.
Right.
That's the problem. What the evidence shows is a lot of deterioration. That's what the evidence shows. And that, like in the presentation I just gave, Stokestack and his colleague say, okay, so maybe there was this organism that didn't need proteins initially. So yeah, you want to believe something that doesn't exist that we can't study. Well, that's exactly what this is. These big beneficial mutations, something that doesn't exist and we can't study. That's like believing in fairies, I mean, I'm just not interested. As a scientist, I'm not interested in that. I'm not. And, I teach a class at Memoria College that we read all the great scientific literature of the past, Archimedes and Francis Bacon and Roger Bacon and all those people. And That's what all of the people who made the science we use today said. You can't just make things up to explain your pet theory.
Right.
You've got to have some evidence. And if you don't have some evidence, I mean, that's all the ancient Greek philosophers did, was they could just make stuff up to explain their pet theory. What makes science different is you can't do that. But somehow evolutionists do that all the time. And that's a problem. So, yeah, show me one of these big beneficial mutations. I'd love to see it. I'd love to study it.
I like that answer. So, in many ways, currently, you're not persuaded. Excuse me. You're not persuaded by that rescue device or mechanism to explain away the genetic entropy problem. That, oh, well, beneficial mutations are counterbalance the damage. It's not persuasive to you, Dr. J.
Not until they can show me. Not until they can show me it happening. You know, and once again, it's got to be in a big way too. You've got to show me this is a mutation that benefited and it somehow outweighed all these other deleterious mutations. Because, yeah, I mean, Michael Behe in his Edge of Evolution book talks about, there are some beneficial mutations that occur. Maybe they're random, maybe they're designed, but they're beneficial mutations. But these beneficial mutations often have very limited effect. And they're often beneficial only under certain circumstances. Like we can have a bacterium can have a beneficial mutation that makes it antibiotic resistant.
Right.
But the way that beneficial mutation happens is usually by deteriorating something in the cell. So now it's only beneficial in that context. You pull the context away, it's no longer beneficial. You've got to show me one of these big beneficial mutations that actually gets rid of some of the effects of the deleterious mutations and isn't something that just works in one situation. And I haven't even seen any of that, you know?
So those beneficials like the antibiotic resistance, the sickle cell anemia, they're beneficial, but it's context dependent. You're saying they're functionally compromising to organisms. Ultimately, I mean, they're reductive. They're due to, you just said, the deterioration of information for a short-term benefit.
So an example, Cipro is a great antibiotic, but there are Cipro-resistant bacteria. And the way they become Cipro-resistant is they degrade their ribosomes. Cipro attacks the ribosome, but it's attacking a particular shape. If we degrade the ribosome, Cipro doesn't know it's a ribosome anymore, doesn't attack it. Now, that degraded ribosome is not even 10% as efficient as the undegraded ribosome, but in the presence of Cipro, it at least makes something. So in the end, the bacterium survives the Cipro because it at least makes something. But you take that bacterium, put it out, and put it in a Cipro-free environment, it's going to be outcompeted by everything.
Right.
Because it's got a deteriorated ribosome, so it can't make proteins really well. And that's the way these at least these antibiotic resistant bacteria, that's the way their mutations work. Now, a lot of antibiotic resistant bacteria don't use mutations. They use genes that are already in the genome to combat antibiotics, because nature produces antibiotics as well. And our antibiotics are cheap imitations of a lot of nature's antibiotics. So a lot of these antibiotic resistant bacteria have these genes to begin with. And we've actually studied this. People have studied medieval poop and they found antibiotic resistant genes from bacteria in this medieval poop. So it was around long before antibiotics because, like I said, nature makes them too.
So I take it then you're not too impressed with the beneficial mutation that's presented. We just had someone in the chat who likes this one. Dr. Dan Stern, Cardinal, Creationist. He's engaged Dr. Sanford and Dr. Carter. So good to have you. He really likes, and others like, the lactose tolerance, the ability. So apparently there's a beneficial mutation in humans that has allowed adults to digest milk. And apparently that's a beneficial mutation that's non-reductive. One, have you heard of that? Two, are you impressed with it? Do you think it's the kind that we're looking for, these super beneficial mutations?
Well, yeah, I agree that nobody's lactose. Well, no human's lactose intolerant. Well, maybe somebody is, but almost no humans are lactose intolerant when they're infants because they're drinking their mother's milk.
Right.
And so they're breaking down lactose then. However, there's a process, and it happens in most Europeans, for example. There's a process where that gene is turned off. later in life because you're not drinking a lot of milk, especially if you don't drink milk, it's going to be turned off. There's a mutation that stops that from happening, right? So in the end, that would be a beneficial mutation if your environment has a lot of milk and you drink a lot of milk, right? And so, but once again, what that mutation is doing is breaking a pre-designed system, the system designed to turn off the gene. Now, we don't know of any deleterious effects that this occurs, but my guess is if your genome was originally designed to turn it off, then breaking that process is probably bad in some way. Now, it may, like the appendix, it may not show up very seriously in a third world country or a first world country where we have all these, ways of fixing problems and ways of treating symptoms. So in the end, whether or not it's somehow has some hidden deleterious effect, it's definitely reductive because it's broken a system that was already there in place. You haven't come up with something new. It's not like there's a new gene that now allows you to break down lactose. You've always had that gene, right? What's different now is it hasn't been turned off. Why hasn't it been turned off? Because the process that turns it off was broken. That's reductive. Right. Now, I can't tell you for sure that's deleterious, but my guess is just like if I see a piece of like a screw fall out of an engine, I'm going to assume that's bad.
Yeah, right. Right. Yeah, me too.
Yeah. So in the same way, if I've got this well-designed DNA system and suddenly some part of it is broken, I'm going to assume that's probably bad. Now, it may not be bad enough for me to notice, especially in a place where there's all sorts of ways to treat problems. But nevertheless, I'm going to assume it's a problem.
Yeah, well, I think that's a good point. And I've heard I believe it was Dr. John Sanford's NIH lecture, which I've got uploaded on the channel on human genetic degeneration, which was a great presentation. And he pointed out, if I remember correctly, even if you just gave the evolutionists 10, give them 20 truly beneficial mutations that are not reductive, it's not enough to counterbalance the damage. I mean, you're going to need hundreds to thousands. I would even say thousands, because Dr. Dan's pushback here he's saying, no, it's not turned off.
And I have to admit, if that's true, I'm wrong because I thought it was actually turned off. What he's saying is the promoter becomes is low efficiently, loses out to other genes competing for transcription factors. So I guess I'd ask Dr. Dan, is it always low affinity, or does its affinity change over time? Well, if it's always low affinity, how is it these other genes start out competing it? Have those genes been promoted in some way or increased in something has to have changed, because like I said, nobody's lactose intolerant as a baby. So we're doing something to either, I thought, turn off. He's saying, no, it's just the way the competition works, which I can believe because I'm not an expert in this. But nevertheless, what changed to make that happen? Because it didn't happen like that when we were babies. So something changed. In my mind, that change is something that's part of the design. And you change, and if
now he says, yes, it's always low. That's the
other metabolic. Yeah. Okay. Other metabolic pathways are suppressed. Okay. So those other metabolic pathways become less suppressed as time goes on. Okay. And I can see that that's part of the general changes that occur from, infant to adult. Yeah. So the higher, when the repression goes away, the higher affinity promoters. That makes sense. That actually does make sense. But once again, is that if I end up that's the designed way it goes. So if I'm able to now digest lactose, I have changed the affinity of the lactose gene, right? So in the end, is that that's still broken the way it's normally going. The way it's normally going is one way. We've changed that. We've broken the way it was normally going.
It's ventured off course from the norm.
Right.
Something that's not the norm.
Yeah. And
I guess it's enjoyable for some people to get to enjoy milk as an adult.
Sure. So, but is it truly beneficial in the long term? I don't know. I'm not sure. And like you said, even if it is, that's one out of that's
One
one
out
out
of
of
many.
out of the hundreds of thousands you need. But I certainly take his point that, and I absolutely agree. He probably knows this better than I do, so I'm willing to concede that point. But I'm still not sure how beneficial it is, and whether by changing those pathways, we aren't affecting something else negatively.
That's a good point too, because how much study would it take to be able to tell? I mean, you need a lot of sensitivity on a DNA level to tell, to discover what potential negative effects are. Because, and I like to point out that even if they had 10 or 20 or 100, the question is that really going to offset the damage being done by nearly neutral mutation accumulation? Because nylon digesting bacteria, I feel like you adequately dealt with that one. That was really good. So the big ones I see,
and
yeah, so I guess you reiterated their points.
Yeah. Let's make that clear. I don't know that stuff really well. And I would like to thank Dr. Dan for correcting me on this, because I don't like to say wrong things. And this was the way I actually read it. So, so I appreciate that correction. I really do. But I don't think that helps make your case. I really don't. Because if it's beneficial, it's a very tiny benefit. And we have to think, is it beneficial in other situations too? Because if we're going to make evolution occur, we've got to have lots of beneficial stuff happening in lots of different places and lots of different ways.
I'm looking it up right now, because the select few and the lactose tolerance one has been an interesting one. The nylon digestion one is a popular one, but again, I feel like Dr. John Sanford and Sal, they've dealt with that one. The Lenski experiment. There's another one I'm trying to think for humans, right? Because because a lot of the math that I see, they're looking at so-called beneficial mutations from bacteria and then extrapolating it to mean genetic entropy isn't a problem for longer lived mammals like humans. But I want to see the best they have for humans. I know we see lactose tolerance. Dr. Dan here, he's done a few debates on the channel. The most recent one, I moderated a debate with him and Dr. Jerry Bergman. And if I remember correctly, one of the examples he brought up, maybe he can let me know here in the chat, had to do with a mutation, which I've heard some say it could be epigenetics, but in humans, it increased bone density in some groups. I'm not sure if you're familiar with that. It made him stronger.
With this one at all.
Yeah, and I wish he could. And another one I think had something to do with being able to breathe underwater longer. He's in the chat, so maybe he can
hold your breath, clearly, not breathe.
Yeah. Hold your breath underwater. Not Aquaman here. That'd be a cool mutation, though. I'd like to. That would be,
yeah. I'm a scuba diver. I'd love that mutation.
Right. Yeah, that'd be great. So, okay. With Lenski's experiment, you touched on it earlier, but how, as creationists, what's the best way to do it? Because I know they point to a few so-called beneficial, obviously the citrate one's the big one, but are those also at the end of the day reductive that you found, Dr. J?
Well, I'm, like I said, the citrate one is a designed one, so it's not reductive. It's actually benefiting, absolutely, but it's designed, and Dr. Minich clearly showed that. But I think what the Lenski experiment does, I wrote a blog article on this many years ago when they were at like generation 45,000 or something like that. And I said, and Lenski's own papers show this. What they show is everything that can happen by evolution has a limit. And they actually have one of them has this great graph that shows the fitness, which is defined by several factors, including how often they reproduce and so forth. Fitness as an amount of accumulated mutations. And what they show is after about 3 mutations, all the rest of mutations make no effect at all in fitness. It's a couple of things that work, and then there's this limit that's reached and nothing else can happen. Nothing else can improve. And that's what the Lenski experiment has shown. And the Lenski experiment to date says that they've made all possible mutations 3 times over in the genome. And yet these guys are still the same species, still the same thing. They admittedly, they've made a couple of new strains, but after 75,000 or however many 1,000 generations are there now in the Lenski experiment, all possible mutations 3 times over, and they haven't produced anything other than a couple of new strains that arguably might be a little better, but that's it. that shows that's exactly what no, I would disagree with that. Linsky mutator strain did not disprove genetic entropy at all. It actually showed that most of these things are neutral or deleterious over time, not beneficial, which is what genetic entropy said. Now, the effect on a bacterium is going to be a lot less because its structure is a lot less complex. So deleterious and neutral mutations in a bacterium aren't going to have the same survival impacts that deleterious mutations in a genetic eukaryotic organism with a much larger genome is going to have, I would think. But the point is, if I've done every possible mutation 3 times over and still can only produce a slightly different strain of bacterium, that tells you that Behe was right. There's a very serious limit to evolution.
Yeah, his pushback is, well, if you have all the mutations possible and yet no extinction in these bacteria.
Yeah, but you don't have them all and happening in one bacterium, right? And they have 12 populations that they continually, they continually change the conditions on and everything. So, first of all, it isn't natural. Secondly, you don't have all those mutations occurring in one bacterium. You have them occurring one at a time in different bacteria. Right, 1 or 2 or 3 at a time in different bacteria, right?
Are those bacterial populations in Linsky's experiment? Are they throwing out a lot of genes for faster replication? Like, is this their way of adapting to that environment? Okay.
No, I don't think they're losing genes or anything like that. They're just having mutations that are mostly neutral or most are deleterious.
Okay. Because I'm thinking back to Dr. Sanford's lecture at the NIH, and somebody in the Q&A brought that up.
Yeah.
And I feel like I remember him saying that overall now that they've shrunk in functional genome size, but don't quote me on.
I don't know that. I have not seen that. I have not seen that in any of Linsky's papers.
Okay.
But I don't read every one of them, so I could have missed something. John Sanford would know more than I.
I could be misremembering. I'll go back, and so don't quote me on that in the chat. He says Dr. Wile just described exactly why GE doesn't. He's coming at you, Dr. Wile. We can pretend this is a debate. He says they all get mixed of mutations, and they are on a net stable. I find that one hard to, but a net stable. So they're not going downhill or uphill. They're just kind of remaining at a point.
But certainly Linsky's experiments aren't saying they're staying stable. They're some of them are losing fitness, some of them are gaining fitness. So there's no stability there. So no, I would absolutely disagree with that. They're not on net stable. I don't think Linsky would agree with that. I don't know. I've never met Linsky, but they're certainly not stable, not according to the papers. So
the biggest objection I see, or wait, especially just in comment sections and students of evolution, they'll say, okay, nearly neutral mutations accumulate, but they're effectively neutral, right? We see that in these papers that no mutation is strictly neutral, they're effectively neutral. And they'll say, well, by effectively neutral means is at first, selection can't see them. They're effectively neutral. Selection is not acting upon them. But eventually they build up so much in a population that the load becomes so large that natural selection or Mother Nature can now see those effectively neutral mutations and then deal with them, rid them from the population, which means, I guess, those mutant individuals are removed. And so that prevents eventual genetic degeneration. I'm curious, in your opinion, is that a good response? I mean, to me, okay, so what, then extinction? Eventually they get to a point where what? Selection removes all those individuals?
Yeah, I mean, once again, the problem is we're working on not a whole lot of data, right?
Theoretical,
yeah. We're working in areas we don't have a lot of data on. The only real data on this is Linsky's experiment, and it's showing that that we're not making anything essentially new. And we're making a lot of bad things as well. And also we're having to keep maintaining the populations. It's not like these guys live on their own. They have to be maintained. They're like, a domestic pet lives a lot longer than a wild pet. I bet a domestic bacterium lives a lot better than a wild type bacterium. But once again, I'm not a biologist, so maybe I could be wrong about that. But my point is we're working on not a lot of data. And I agree that there would come a time, I think, when any gene gets so mutated that if it really is an essential or a big factor in survival, then natural selection is going to kill it off. And those people who didn't harbor all that big accumulation of near neutral mutations, they're the ones who are going to become the dominant version of the population now. I agree with that, but I think the point is not necessarily that we're all going to die because our genes are all deteriorating. I think the argument is that our genes are deteriorating so much that natural selection has a hard enough time weeding that out. How could we get beneficial mutations on top of that? There's I can't remember, it's called the Red Queen hypothesis, and that is our genes are deteriorating so fast that all natural selection can do is keep that at bay. And so natural selection is always running, but getting nowhere. That's the Red Queen. So that's one possibility, but that's what I see. I see the rule in genetics is deterioration. It is not producing great new beneficial mutations. We can point to maybe one or two that are partially beneficial in some situations, but no, the general rule is deterioration. I'm not sure how anybody can argue against that. So the argument is, how can I get this increase in information in a pool that is seemingly just generating more than anything else? And that one argument, the big beautiful the Trump big beautiful mutations would have to overcome that quite a bit.
Yeah, Dr. Dan talks about the Red Queen is the idea that in competitive relationships, reciprocal selection prevents stasis.
That's another version of the Red Queen hypothesis, but there are lots of them, because in the end, it's just this idea of running somewhere and getting nowhere.
Right.
Yeah.
Well,
running and getting nowhere.
I think you explained it well. I mean, genetic entropy, from my understanding, is just the fact that every generation is a little more mutant than the previous one. Not that we're just all going to go extinct in a few generations, and I think you just said the rule is deterioration. I heard Dr. Rob Carter, he gave a good illustration for this. I think he said, take a room of 100 people. Everybody's multiply mutant. We're all more mutant than our parents and our grandparents. Maybe a few of them are a little more mutant than the rest, but everyone's still mutant. Okay. So remove 50% of the most mutated individuals in that room of 100. Okay, fine. So you remove them. You're still left with 50 people that are more mutant than the previous generation. Yeah. So sure, selection can act as these nearly neutrals accumulate and can remove some of the most mutant individuals, but it still doesn't solve the issue, does it? Because the rule, like you said, is deterioration.
Yeah. And you know, that's where genetic entropy is awfully hard to argue against, because we can just track mutations that have occurred that have not been weeded out by natural selection. And like you said, every person is more mutated than the previous generation. This doesn't mean we're all going to die. But what it does mean is deterioration of the genome is the norm. And so somehow, since evolution is the opposite of that, we have to figure out, if you're an evolutionist, you have to figure out how that happens. How is it that evolution can go against the norm so much? And I think that's something that I haven't seen a satisfactory answer to, certainly.
I think it's a great point. So as we move to the 2-hour mark, I wanted to sneak in a couple questions based on criticisms that came in and then we'll kind of end with a bang. But to leave no stone unturned on the junk DNA issue, I think you did a great job just dismantling all of the objections and responses. One of them that typically comes in is, okay, so 60 to 80% of the genome is transcribed. Or what's the best way to iterate that? Because 80% activity, but is that all? Is not all that 80% is transcribed? Is it, Dr. J?
Makes it out of the nucleus.
Okay. So
it's got to be. It's in mRNA form now.
Okay. So, and like we said earlier, that should be beneficial activity then, because transcription's an energy intensive resource, as you said, intensive process. I know Dr. Dan has problems with that, because they'll say, well, a lot of what we argue are now functional, like pseudo. William Lane Craig, he put out a video and said, why I believe humans and chimps are related, why I'm a theistic evolutionist, is because shared pseudogenes.
Yeah.
Well, I mean, you answered that earlier. A lot of those are actually functional. Some of them are truly broken genes, like the gulo gene. But they'll say some, like some ERVs, some pseudogenes within that activity are what are called transcriptionally silent. So actually don't produce an RNA, and there's a lot of those, and so those must be junk. And basically the question is, are these transcriptionally silent DNA units, are they actually junk, or is there another use for those too?
Well, Peter Borger several years ago wrote 4 papers on this, and he called them variation inducing genetic elements, VIGEs. And I think he spells his name Pieter.
Yes.
Yeah. And so he said that's what these guys do. What these guys do is and we see them jump. You know, they go to different places in different people's genomes. So they are moving around. So that indicates they're doing something. And he says what they are is they are the agents of these design mutations. He didn't have the evidence of Scott Minich back then, but he said, look, the genome is clearly designed to change and designed to adapt. And these, what we call ERVs and so forth, are actually the agents by which the DNA does adapt to different situations, does follow that design. So no, I would say transcriptionally silent doesn't mean non-active, but I think it's awfully hard to say transcription, it doesn't happen, or transcription would happen on something that is silent. Because the ERVs aren't silent. We see them doing something. We see them moving, right, about in the genome. So they're not silent. They're just not transcriptionally active. They're doing something else, because we now know it. You know, it used to be thought that the only thing was the sequence of genome, a sequence of nucleotides. That's it. That's everything about DNA. And just like junk DNA, we know that's wrong now. And in fact, the structure of the DNA makes a huge difference. How easy it is to get to the gene makes a huge difference in how quickly transcription occurs and so forth. So the actual physical structure of how the DNA is packed and how it is unpacked makes a big difference. And these ERVs could be affecting that, because we know they're doing something because they're moving around.
Oh, and they're doing so much. I mean, now there's papers 20, 22, 23, 24, which talks about how ERVs function in the immune system. They work to fight off bad viruses. They're necessary for embryological development. I have one paper that I show in a talk where in the conventional literature, it literally admits if we did not have ERVs, we wouldn't exist.
See, now you're much more knowledgeable about this than I am. I didn't know about this at all.
Oh, it's gotten even better. They perform viral mimicry, because now cancer researchers are looking to ERVs to study cancer therapy drugs.
Oh, wow.
Because they could be turned on in a tumor cell, and then the immune system, mediated by the p53 proteins, like, whoa, what's going on here? You know, because it's mimicking a bad virus, and so they destroy those tumor cells. Viral mimicry, and it's incredible. So yeah, and then even the transcriptionally silent pseudogenes or ERVs are said to have function. And I'm just navigating the chat here. Okay, I really wanted to get this in, Dr. J, and then I look forward to hopefully more presentations with you, because you generate some good discussion in the live chat, brother, but then also in the comment section. So I put out a video after our previous show on C14 and dinosaurs, and one critic over Facebook, I see this with reasons to believe a lot of the old earthers. They asked this: why is it so with the C14 that we find in dinosaur fossils that really shouldn't be there if they really are millions of years old? Because the decay rate is pretty fast in C14. So the question is this: why is it always trace amounts? If they were only 6,000 years old, where's the rest of the C14? The half-life is 5,700 years. Why isn't there 48% in it, and then the critic continues, but basically, why is it only trace amounts?
It's not. That's the thing. That's absolutely false. We can carbon date dinosaur collagen and bone, not collagen, just bone. We can date it to 29,000 years. That's not trace. That's halfway to the theoretical limit. So that's really basically 50% of what could be there is still there. That's the way you get 29,000 years. So it's not trace. All right. Now you could argue, if you argue that there's some natural process that's making this come up with 29,000 years old, then every carbon date that says 29,000 years old or older has to be brought into question. And that's a huge part of the literature. It's just false that it's trace amounts. There are trace amounts in like diamonds. I agree with that. Estrogen Genesis does a lot with carbon 14 in diamonds. I agree that's trace amounts. I'm not sure that's really there. And if it's there, I'm not sure. I'm not sure it says anything about the age of the Earth. But in dinosaur fossils, it's not trace amounts. It's up to 50% of what could be there. That's not trace. Right. And it doesn't really work that way. It's not linear. But what I'm saying is you're only halfway through the possible date range. So therefore, if you really think that contamination or something like that can explain these numbers, then you have to call into question all of the carbon 14 dates that are 29,000 years or older. Because if I can believe that I can contaminate a sample to that level, then I can't believe that any of those other samples that date that old aren't contamination. And I don't think anybody's willing to do that. And so once again, it's not trace amounts. It's serious. And I think I said this before in the talk I gave. I'm okay if you really think these guys are millions of years old, but you've got to study this issue to figure out how the carbon 14 levels can be so high.
Right.
And if you aren't doing that, then you're not participating in science. You're participating in religion. And so to say trace amounts, you're just giving a religious argument because that is factually incorrect. It's not trace amounts. If you call that into question, you're calling into question a huge part of the carbon 14 dating literature because lots of them go 29,000 years or older.
Wow. And I'm just so flabbergasted that we get so many comments, even from older scientists that continue to say that. They'll say, well, those, and I think you just refuted this, those trace amounts, which you just demonstrated are not trace amounts. So it's an assertion or religious belief, equal the in situ and contamination. But no,
and studies show it's not contamination, because if it was contamination, the carbon 14 concentration would be higher on the outside than on the inside, and it's not. It's random, right? Yes, they do. First of all, they do account for possible contamination. There's a whole protocol you use to reduce the contamination effect for all samples that are carbon 14 dated. And also, in the end, the folks who do the dinosaur studies have done, right at the edge of the fossil, right on the interior, not right at the edge, but near the edge of the fossil, in the interior of the fossil. If it's contamination, it ought to be higher on the edge and lower on the interior.
Right.
And there's no pattern. In some examples it is, in other examples it's not. So in the end, you've got to at least be honest. And I have no problem. Try it. I would love to see someone come up with a method that shows me how that much carbon 14, because it's not trace amounts, can actually be in a dinosaur fossil. I'd love to see it. And I think the other side of the argument is, why don't they date as 4,500 years old? Because that's what, roughly the flood probably occurred on. They don't date as 2,500 years old because the environment pre-flood was radically different from post-flood. It's the atmosphere interacting with cosmic rays that makes carbon 14 to begin with. There was simply less carbon 14 pre-flood. So these things look abnormally old because they're starting with less carbon 14 in the environment.
And you're looking at this quite objectively. Well, firstly, you said in your presentation,
nobody's objective.
Right. Well, I guess I say objectively because, so maybe it's the wrong word, because you've pointed to diamonds, which, I kind of like the diamonds argument, but you're even kind of giving
that's where it's
out to the critics.
Sure.
You're saying, well, maybe that's trace, but the dinosaur fossils are definitely not trace amounts.
Yeah.
And
yeah.
that chapter.
I'm trying to be scientific, and this is where these kind of discussions rarely are scientific. They can be, they can be saying, "Oh, I'm an objective science." Nobody is, and so first of all, if you say you're an objective scientist, you're lying, because nobody is. All right, so everybody brings in their biases and so forth to begin with. But secondly, I'm just acting as a scientist. I'm saying, look, I don't believe any of this stuff is set in stone. I'm willing to change my mind. I've just got to get the evidence. Now, there are probably some things I couldn't change my mind on. I've seen the effect of Christ in my life. I couldn't change my mind on his existence and his devotion to people and all of that. But I could easily change my mind on the age of the earth, because I don't even think it would affect my theology significantly. It would have checked the first 14 chapters of Genesis significantly, but it wouldn't affect my other theology in any really significant way. I could give up that. I just, as a scientist, I see so much more evidence for a young earth than against it. But I'm willing to look at any evidence you got for a old earth. And I'm willing to, and certainly any evidence you can give me on how there are not trace amounts of carbon-14 in dinosaur bones. I'd love to hear it.
Well, I like that. You can look at the data like a scientist. And I answer because you made a good, there's a couple things I can't change my mind on, because I've seen that we've been enlightened on the truth of Christ, the gospel. You know, I like to say, well, at one point when I was a kid, I believed in Santa Claus. Then my parents told me there's no more Santa Claus. I was enlightened. Am I one day going to go back to believing in Santa Claus? Probably not. And so once I received the truth of the gospel, you know, there's no going back on that. And so, yeah, no, I think that's a good point. Okay. I want to wrap it up with this now. So here's the last question. Dr. J, I always appreciate your time. You're an encyclopedia worth of information. We got a lot of good clips from here as well. We had one commenter earlier say, I think it's sped by, oh, here it is. It's talks like this is why I support. So people like these interactive talks where we can, like you do, you present, but then we also get to engage the critics in the chat, which I have fun doing. So critics on dinosaur soft tissue. That was one of your arguments in your last program. I put out a clip on that as well. They argue that, I'm sure you're no stranger to these, that iron preservation and mineral encapsulation can stabilize proteins for very long periods of time. How strong is the chemical evidence that these tissues are actually original biomolecules rather than mineralized structures, Dr. J?
Okay, the fact that they're biomolecules, we know for certain, because there are certain microscopic stains that only bind to the molecules. They don't bind to fossilized, mineralized, patterns. They bind only to the molecules. And when you put those stains on these tissues, they bind to the molecules. And so we know they're real molecules. Also, there are papers. I wrote a blog article on a paper that actually sequenced the amino acids in the proteins, and showed that you can't sequence the amino acids in the proteins if they've been mineralized, because the way you sequence it is by using the chemical properties of those amino acids. So we know they're not mineralized, and I don't know any knowledgeable person who says that anymore. I really don't. Now the other one was what? Oh, for iron. Yeah. So this is Mary Schweitzer's argument. Mary Schweitzer did some experiments with ostrich blood vessels and used a very iron rich, very unnatural source of iron, and showed that the blood vessels after 2 years that were exposed to this very unnatural source of iron were more preserved than the blood vessels that weren't exposed to this unnatural source of iron. And her experiments are valid. I don't see how 2 years of experiments can do anything for a fossil that's been under the ground for 1,000 years. But okay, because as I said in my other talk, I'm very careful with my extrapolations. But here's the overall problem with her idea. When you actually examine chemically these structures, you find very little iron. I mean, once again, they sequenced the proteins that were found, and they didn't find any iron embedded anywhere in the matrix or anything like that. So it's possible that iron can preserve things, but we don't see it in these soft tissues. And if you're going to tell me these soft tissues are preserved by iron, I want to see the iron, and I don't see it.
Wow. Okay. So So I'm taking notes too. I'm learning from you all the time, Dr. J. So iron could hypothetically preserve some of these soft tissues, but you're actually pointing out that they're not really finding much iron in the soft tissues that it's supposed to be there to preserve.
Yeah. Yeah. Now, there are sometimes you can find iron-rich soft tissue. Like, for example, there are pictures that some people say are blood cells. They're not really blood cells. They look like blood cells, but those are rich iron sources, and they give the red color. But those are only certain structures, and they're usually associated with certain types of blood vessels. But when you look at these nerve cells that are preserved, you look at these bone cells that are preserved, and these bone cells aren't mineralized. I mean, the Dinosaur Soft Tissue Research Institute has a video of somebody using using pressure on the slide to flop the molecule or the cells around. Mineralized cells don't flop like that. But you don't find iron in these samples. So you find iron in a few places where you expect iron to be. You don't find iron in these soft tissues where you don't expect iron to be. But if you're using that as your preservation mechanism, you got to have iron everywhere.
Interesting. So as creationists, when we go out and defend the faith, give presentations, talk to people, what's the best simple way for us to accurately reiterate this argument? I saw a post, an important post by Dr. Mark Armitage, who is
he's not a doctor, but yeah, he does the serious scientific. I'm not disparaging him at all. He's an excellent. He makes a microscope sing. He is an expert on them. He does it better than anyone I've ever known, and I've known some high level biologists. But he isn't a doctor. We got
to and that's helpful.
Yeah.
So Mark Armitage, he was encouraging us as creationists to accurately explain the argument. Where, like, sometimes we'll say red blood cells, but it might not be red blood cells. Okay, so as creationists, what's the best way for us to explain that argument? What is the soft tissue, but what is proteins, collagen? How can we explain it to people where we're accurate?
to me, the most accurate thing, because we know this for certain, is there are soft cells. They're not alive anymore, but they are cells and they are soft. And so we can take this soft tissue and take it down to the cellular level and get soft cells out of this soft tissue, right? So proteins and all that, that's all really interesting. But for a cell to be preserved, but not mineralized, to be preserved as soft, indicates very little deterioration. Because, it's nice to find proteins, but proteins can be left over after the cell deteriorates, right? The cell being there and being floppy, and it's not just Armitage that's done this. Armitage has done the best work on it because he's actually pulled the cells out of the matrix they're included in and all of that, and shown that they were floppy. I don't know of anybody else who's done that. Somebody may have, but there's plenty in the secular peer-reviewed scientific literature that shows these cells that and the authors say they aren't mineralized, but they're still but they haven't extracted them out individually, but they show cells that are there and they're soft. So one thing we can say for sure is individual cells are not mineralized and yet are there and they're soft. Wow. And so that indicates very, very little degradation.
It's awesome.
And once again, these cells aren't covered with iron.
Right. Yeah. And exactly. So they're not covered with iron. Soft cells are present, not mineralized, not deteriorated, which you'd expect them to be after millions of years. Are you familiar with Well, I like what you said. It's not just Mark Armitage. I mean, we had Dr. Brian Thomas, who just recently, I think it was in Analytical Chemistry. Yeah. He published a paper with other authors on another soft tissue finding in the conventional literature. I mean, there's Creationist publishing, but he gave a talk here where he went over, and I can't remember every example, but it was about 120 to 140 different findings. Yeah. And then you presented the same thing in your presentation last month or two.
Yeah, I can't remember. Did I do the scrappy bones? Was that in there? I think that's my favorite thing, where these guys just want to find out how common is this. So they pulled what they called scrappy bones from the museum's warehouse or museum storage facility. He said, we don't even know what dinosaurs these bones came from. And like out of the 9 bones they looked at, 5 or 7 of them had soft tissue in it.
Wow, that's incredible. So they're finding more now that they're actually looking.
Yeah.
They're finding more.
Oh, yeah.
Where before, like with Mary Schweitzer, because I feel like some people that are not up to date critics, they always just go back to Mary Schweitzer, but there's so much more that's been discovered since her.
Yeah, and she obviously, and she's a Christian, but not a young earther, and she doesn't like
Right.
young earthers using her stuff. So let's make that clear. That doesn't mean, I mean, I'm sure a lot of people didn't like Einstein.
Well, they always remind us of that one, Dr. J.
Yeah. So, but in any event, she, held the ground for a long time for all of these old earthers who once again turned on a dime. Initially, there's no way that's soft tissue. Soft tissue can't live for millions of years. And now when you do all the chemical analysis, you find it on every bones. All the old earthers have turned on a dime and said, well, it's not so surprising that they're soft tissue. And once again, this is what evolutionists do. They turn on a dime. 1 minute, it's great evidence for their theory. When it's debunked, well, you don't expect it anyway.
Right. It's yes, an answer to contradictory questions is just yes. Evolution's true because the sky is blue and the earth is round.
Yep, exactly.
Have you heard the criticism towards Mark Armitage? It's a triceratops horn, right? That he's looked at with the
he's looked at so many. That's the thing. I remember there are these three people on YouTube. One of them doesn't even want to show his face, so he uses some dinosaur model. And they said, "Oh no, this is a rhino horn."
Yeah,
but
bison.
Yeah, but bison horn. Sorry, not a bison.
No,
horn.
yeah.
But he's found this in Nanotyrannosaurus fossils. He's found this in other, and he now has a sort of a roving lab where kids can come use his telescope and find dinosaur cells, so it's not even very hard anymore. And it's not just that one triceratops bone, it's multiple fossils from multiple different samples and more than one species.
Wow. And it's reasons like this that I say it's a great time to be a biblical creationist, because when the best critics and oftentimes scoffers like those attacking Mark Armitage.
So like if Dr. Dan says that's a deal breaker by Bison Horn Armitage, please actually look into this, Dr. Dan, because you're completely wrong. It's not just one horn, it's not just one sample, and it's not just one species. All you have to do is go to dstri.org and you can see it all. And he's published in peer-reviewed microscopy literature.
Yes.
And it's from different species in the peer-reviewed microscopy literature. So please look into that, because I'm willing to admit I'm wrong about the lactose thing. You should be willing to admit you're wrong about that.
In science, you have to, and it's all about model building and how do you get to the best possible model and understanding if you're not going to alter your views and admit when you're wrong. And you said Nanotyrannus, right?
I don't know. It's a small version of Tyrannosaurus.
I think you're right.
I don't know. I don't know much about those individual, and a lot of dinosaur species change over time because, I was taught about Brontosaurus. Turns out that was a mistake.
Right.
So all these things change over time. So I think it's called Nanotyrannosaurus, and it's a small version of the Tyrannosaurus, but that's one of the other species he's found. And he's found nerve tissue from that species and all sorts of stuff. And so, you really need to look into that before you start saying false stuff like that. And like I said, I'm sorry I said false stuff about lactose, and I'm glad you corrected me.
Well, that's what I love about your previous talk. So I want to encourage, as we wrap up here, to the audience, please, because I know a lot of people are enjoying you. Again, Dr. J, I appreciate your energy. You're well presented always. You're up to date on the science, which is what we need here in 2026. And I want to encourage people to check out your previous talk, Why This Nuclear Chemist Believes in a Young Earth. You came to young Earth creation because of the science.
I wasn't
older.
older.
We get a lot of critics that say, oh, the only reason there's even young Earth creation scientists is because, the Bible told me so. So nothing's going to contradict that. You came at it from the scientific angle. And the evidence that you use, here's the point I want to make. The evidence that you use in that talk are just some of the strongest lines of evidence for young Earth creation, that the best rebuttals or rescue devices you got great answers for. And those are my favorite pieces of evidence, Dr. J, the ones that can be defended, that are defensible. Okay, I want to hand it to you. Any final words, final thoughts you want to leave us with? I really appreciate you giving us two plus hours of your time, Dr. J.
Well, this is really fun. When you said coming to the 2 hour mark, I was like, really? Wow.
I know, it just flew by.
So that's fun. No, I really just think that anyone who's trying to seek what's true, whether you believe in a Christian version of truth or some other version of truth, anyone who's a scientist has to believe there's things that are right and things that are wrong. And so I think you need to look at both sides of the issue. That's how I came down as a young earther. I thought I was an old earther. I originally was an atheist, and it was science that changed my mind on the existence of God, and the science that changed my mind on the age of the earth. And, like I said, on the existence of God, I'm pretty set, but I could be wrong on the age of the earth. I could be wrong about a lot of things. So just don't take my word for it. Look into it for yourself. That's how you educate yourself. And not everybody can learn everything about everything else. But if the age of the earth is something you think is really important, then look at all sides. If genetic entropy is something you think is important, look at all sides. That's the key.
Just out of curiosity, because I've got Hugh Ross's new book here, which we've examined on the channel.
I haven't read that one yet.
Okay. Yeah. So we've examined it with some speakers. From what you've heard, let's say the science did come and something came, but what you said during the presentation, creationists are making testable predictions, junk DNA, vestigial organs, other things, so that's the gold standard of science. Yeah. But if something did come out that really contradicted the young earth creation position, you said a couple things: the essential issues, salvation, the deity of Christ. These things are not affected by the age of the earth. But would it be difficult for you to swallow some of the arguments that we see from Reasons to Believe, to argue that the Bible teaches, especially in Genesis? Because I see a lot of these arguments for a local flood, and it's not convincing to me,
Yeah. So I know Hugh Ross's progressive creation and Gaussian distributions for days and so forth. That would be impossible for me.
That'd be tough.
If I became an old earther or a theistic evolutionist or any kind of combination, I'd have to go where C.S. Lewis went. That the first 14 chapters of Genesis are myth. And myth is not necessarily bad. Myth has a negative connotation to us, but myth is not necessarily bad. C.S. Lewis says there's an enormous amount of truth in myth. And what these 14 chapters are a myth that teaches us something important. I would have to go there, because there's no way that Hugh Ross stuff is reasonable in my view. I just can't see it.
Yeah, me too.
Gaussian distributions of days and only a local flood, and no, that's too much.
And I haven't seen good counter rebuttals. Well, even theistic evolutionists like William Lane Craig. I mean, he's gone with the mytho-history point. But I'm sure if he was convinced by Hugh Ross's arguments, he'd be one of the first. Why not? He's an old earther. But a lot of old earthers are not convinced or persuaded. Progressive creationism is not very persuasive.
Not to be, certainly. And the gap theory is even worse.
Oh yeah. I mean, that's just old news. It's hard to believe some people still use that. Well, Dr. J, it's been fun. It's been a blast. I look forward to hopefully you being willing to be seen with me in public for another couple hours in the future. So
definitely, it's too much fun. But yeah, I'm getting hungry. I do have to go eat.
Okay, good. So you've got great endurance. So this has been a lot of fun. To the audience, thanks so much for tuning in. Stand for Truth is signing out. God bless you all. We'll see you soon.